I now call Professor Andrea Balsari, who will speak to us about microorganisms as possible therapeutic agents. I would like to thank the organizers and Professor Lanfranchi for having invited me to speak here. My paper here today is a little bit external to the core of the discussion here today. What I'm speaking about are the bacterial components in terms of possible treatment, anti-tumor treatments. I deal with possible alternative types of treatment for the oncological patient. What correlation is there between oxidative stress in the oncological patient and the use of bacterial components for the oncological treatment? Well, these are two sides of the same coin as I see it. Most inflammatory processes are of an infective nature. The inflammatory process of an infective nature leads to a process that can lead to cancerogenesis and hence to the progression and the growth of the tumor cell, but it can also determine a response of the immune system. This response might lead to the development of a protection against this cell. And this had already been seen many years ago. At the beginning of the century, at the end of the 19th century by Coley, who had often seen in terminal patients that there was an infective process which could lead to an improvement of the neoplastic symptomology. After having made this discovery, Coley also tried to inoculate live bacterial layers into the patient, obtaining some sort of success, but the side effects and the actual problems that there were in that day and age led to the termination of this type of therapy. Up until the end of the 1990s, that is to say 15 years ago or so, we didn't really know how our immune system functioned, so that we could recognize the various types of microorganisms. We knew everything about the antibodies, about the lymphoreceptors, but we did not know how our immune system can distinguish between one microorganism and another. We must have a completely different response if we are attacked by a streptococcus rather than by the larva of an insect or by a virus, and how the immune system could understand this difference and develop a different response, well, that was unknown. Almost by pure chance, around the end of the 1990s, a number of studies were carried out on the Drosophila. This is a mosquito which has mutations, and by carrying out such studies, we noticed that the Drosophila who had alterations according to certain genes, they were much more sensitive to mycotic infections. Hence, we analyzed what this gene TOL produced in the Drosophila, and we saw that it produces an antifungal activity. Now, this remark of the end of the 1990s led to examine whether in man, too, there were genes similar to this type of TOL gene. We found 10 genes in man which were similar to these TOL genes. However, they have a different function, a different effect than those of the Drosophila, in the sense that they are not capable to produce antifungal or antibiotic activities, but our organisms can recognize through them what kind of microorganism has penetrated it. These genes acknowledge sequences that are present in the various microorganisms. They have a polysaccharide, for example, gram-negative RNA present in viruses. Flagellin is present in other microorganisms. So on the basis of the cells that recognize the various components of the microorganism, our immune system is capable of understanding what type of infection has attacked it, and a response will be provided with immunoglobulins of class E, or the activation of macrophages if it's a gram plus bacterial infection, and so on. The discovery of these toll genes also led to the production of synthetic molecules which reproduce the molecules of the various microorganisms capable of activating a specific toll, while coli, by inoculating bacteria as they were, determined a complete reaction. That is to say that a very large number of different tolls were activated. If we have specific molecules, synthetic molecules for a given toll, well, then we can activate that toll-like receptor. In oncology, therefore, after carrying out several experimentations in animals, we started by using a small sequence of 20 nucleotides which are present in the DNA of bacteria, but not in the human DNA.